So I took on the task of then approaching the importance of these traditional epidemiologic studies in the context of these fabulous other kinds of designs that we've heard about so far today. And I hope that I leave you at the end of this with the notion that it's really a great opportunity that all of these different kinds of studies, from the hundreds of thousands to what I'll show you in the hundreds, really have a place and give us different information. And I would love to hear the discussion that comes at the end of this after presenting. And I just wanted to show you guys this because I think it sets the context for what you've heard already and for what I'm about to say. This is actually a figure that we use in the Department of Mental Health to think about how mental health and public health overlay each other. But I think it works for the work on autism that we do in the center, and certainly the kinds of things that you've heard today. That some of the talks really were up here in terms of the prevalence and descriptive information to some of the causal questions. And then you've heard just now about some of the services and policy kinds of questions. And so when I talk about all the different kinds of designs, our group really sort of hangs out mostly in this quadrant, but I want you to be thinking about the studies big and small from this whole perspective and not so narrowly focused on just causal kinds of projects. But as I said, our group, my group specifically, works more on these causal kinds of questions. And we really come from the perspective that if you want to understand the causes of autism, and this goes back to the very beginning talk today, that you really cannot ignore the overwhelming evidence that genes play a role in risk for autism. But you also cannot ignore the evidence that they must not be the only explanation for what causes autism, and that really it's by joining the worlds of environmental epidemiology and genetic epidemiology and environmental and genetic medicine that we can try to understand these underpinnings. And as was discussed, in some sense, some of these are now getting easier to answer, and these are still quite challenging. And a lot of the decisions about study designs have to do with thinking about how do you maximize your opportunity to learn something about what's going on the right. And our group really tries to put these together and think about how genes and environment might work in connection. And so the designs that we presented today, and I loved, Diana gave a version of this just amongst the registries idea, not even thinking about some of these other designs, about how, when we move from these studies that I'm going to talk about at the bottom, cohort and case control kinds of traditional, go out and actually recruit and consent somebody and interview them or take their blood samples or whatever, up to billing databases and then ultimately mandated national registries, we're moving for sure from tractable sample sizes that are in the hundreds to thousands up to sample sizes that are in the hundred thousands to millions, right? It's just a very different world in terms of that sample size. But we do pay a cost, and Diana already mentioned this, the thing that the sort of flip of that coin or the yang to that yin is that we also sacrifice something in terms of depth of information. Whether that information be about a phenotype or what kind, what the presentation looks like in a child, or the information be about, and what you just heard about, what the socioeconomic status or racial background might be of individuals. There's all kinds of depth of information that you lose when you make these kinds of different choices about design. So I'm going to talk about these traditional sort of primary data collection options. I also want to be clear, when Dr. Krohn talked about some of the case control and cohort designs within Kaiser, it's a tough thing for her to talk about because she's done case control and cohort designs that still didn't touch and consent particular people. It just said, amongst this data that I already have available, I'm going to identify cases and I'm going to identify appropriately matched controls. You can do this, bless you, the same kind of thing in national registries. Those are indeed case control kinds of designs. What I mean, though, are the ones where we're actually then identifying those cases and then going out and recruiting them and trying to touch them in some way and talk to them and have them consent and those kinds of things. So that's a subtle difference. And if you don't mind me going through a couple of slides of kind of, I feel a little bit more didactic, and I hope that I'm not appearing to speak down to anyone, but I think it's helpful, at least for me, to always remind myself of the differences and the trade-offs, and I think graphically. So I just wanted to kind of show you, when we're starting to think about these case series versus case control versus cohort, and then something that I will propose, enriched risk cohorts, what are the differences in terms of your graphical thinking? So, I have this vision of people are conceived, and then they're born, and then they grow to age 1 and age 2 and age 3, and that's kind of what I'm showing by these lines. And along the way, some of them receive an ASD diagnosis, right? And so that's what's going on here. And that's ultimately what we're trying to capture with all these different designs, right? Often when we think about the first pass kind of thing, whether it's that we touch them or it's just administrative or other kinds of data, the first thing you can do is cross-sectional. Just say, okay, in 2013, I'm going to take a look and see what I see. And so if you did it for this graphic, you'd have those orange ones are ones that then you consider to be cases, and the blue ones are the ones considered to be controls, and you can ask whatever kinds of questions you want to ask about those. Typically, we don't stop there at just looking at one point in time. Often, and I should just say, in the case control setting, we can look at both those who were and weren't diagnosed with a particular disorder. Often we want to do more than that. And sometimes to answer more than that, we want to focus on maximizing our ability to find those orange cases. So we've heard a lot about the prevalence, 1 in 68, 1 in 53. Those are shocking numbers in the context of where we thought prevalence of autism was 10 years ago, but they're still relatively low numbers if you're talking about sampling 100 people. Because if you sampled 100, if you sampled 68 people, you might only have one case, right? If you sample 100, 1.5 cases, something like that, right? So it's still a relatively rare disorder. So one of the sort of mainstays when you learn epidemiology is if you have a rare disorder, sampling on cases is efficient, because otherwise you don't get enough cases to answer relevant questions, right? So that's why this kind of case series or identifying cases in a population is critical and is a major tool in autism, and why we started today hearing about, if you really want to go after these things, try to capture people who are already affected, because that's an efficient way of going about learning. And then you can ask sort of what happened before and what's going to happen next in those samples, right? So that's where this sort of case series idea comes from. And so you can accumulate very large samples, and even for rare disease, this can be an efficient approach. And when I say sampling on cases in this case, I don't mean that you don't have other individuals. You heard about the trios and in the Safari Initiative and others. Often the sampling is by case, by identifying cases in terms of who you see in the population, but then you ask their parents or their siblings or their family members to participate with you. So you can get some kind of comparison or control folks, but you're really reaching into the population by this case identification. Case control studies, it's sort of a similar idea, case identification, but then you think about who's the appropriate control group, and so there's like some toughness in figuring out who that is. I'm going to tell you about a study where we think of two different kinds of controls, those that are typically developing children that are matched on something, but there may be other kinds of groups like non-ASD but developmentally delayed children. That might be a relevant kind of comparison group. There's things you can think about. So that's one challenge when you do it this way, is who are those blue lines that you want to pull from your population? And then the other tough question is, you're necessarily grabbing folks who have already been diagnosed. That's how you know they're a case, right? So everything that you might want to ask, so you can get large numbers doing it this way, but everything that you want to ask about what happened previously is going to take some kind of retrospection, whether it's looking in you know, EMR or med records data, or actually asking people to recall, or you know, some kind of retrospective information. And if it's asking people to recall, you for sure are open to things like recall bias. Moms of children who have a particular health problem are going to potentially over-remember or be more accurate in their memory than moms who weren't thinking about that every day for the last 3 years of their child's life, right? And even if you got that measurement information right, the temporal ambiguity. So this is when this person got diagnosed. When did their autism start, right? Did it start right here? Did it start right here? How about here? How about here? And so when you think backwards, those things get a little bit challenging. Ultimately, you'd want to know, or at least I would argue ultimately, what's happening here at conception, or potentially here at birth, things like that. And that information bias, just not necessarily the recall itself, but how you measure, how good, even if there was no bias, even if it wasn't that moms of autism or families of autism children remember differently than families of typically developing children, even if they had the same amount of mismemory, that misinformation or that information bias is a problem when we do these kinds of case control studies. And I like to think of it as, you know, the sort of cloudiness. If you ask me to think about what happened around birth for my 4- and 6-year-olds, I'm worse and worse at that every year, you know, for sure. So that's the argument in epidemiology writ large, and certainly is true in autism epidemiology, for starting as soon as you can, for thinking about a cohort or something where you're starting before you know about the outcome of individuals in the study. You no longer have this temporal ambiguity. You no longer have this temporal ambiguity, you no longer have this recall problem. There are lots of advantages. And then you follow and see what happens in terms of who gets it. And so I should just say, so that seems beautiful. And I do think that's, we teach that. It's a great study design, thinking about moving forward in time and all the things you learned about cohort studies. The big problem with that for autism is if we did this for all of the, whatever, I think it's like 20-something lines on this graphic, and we follow them till age 4 or 5 or beyond, these are how many orange stars we'd have. And so we follow another set. And we spend all the resources and stuff to do that on this. Still no orange stars. So we could go about that all day long for a lot of people and maybe get one. So you can design a very beautiful cohort study that has all the things that you want, and I'm going to talk to you about some of the things that I think you can get from doing these, but it's not a very efficient approach for something that's still relatively rare, like autism. And so that's if you could think, though, instead about some way of improving the amount of children that might have outcomes that make sense to you, you could sort of increase that efficiency, right? You'll sacrifice some generalizability maybe, and we'll talk about it at the end, but you'll definitely increase that efficiency, and if you can maintain internal validity, this is a reasonable approach. And so one way of doing this that's been discussed in literature is looking for folks at enriched risk for things like family history. You've heard a lot from every single speaker today about how family history is a risk factor for autism. And it doesn't even have to be family history of autism, actually, but family history is a risk factor, right? So if I followed only babies that were at some kind of increased risk because of family history, I might have 2 out of those. And that's because instead of the 1 in 68, estimates right now are somewhere around 1 in 5, about 20% of children who have an older sibling that has autism are likely to end up with a diagnosis. And so that's a very different prevalence, right? So that makes it a little bit tractable to follow in time. You're still going to follow a lot of children who don't develop the disease, but that's actually kind of an interesting thing, right? Because now in this enriched risk group, for that one group that I started with here, I have both cases and controls. So I have sort of a built-in comparison group. Everyone was at high risk, but some ultimately have outcomes and some don't. So that has been the sort of mainstay, and it will still take time. 2 out of that group, 2 more out of that group. If you start accumulating these kind of enriched risk sets, you can do something that that is efficient and can still answer causal questions. And it doesn't have to be just enriched risk because of genes. It could be because of babies with preterm birth. We saw a lot about preterm as a risk factor. We saw a lot about how advanced parental age is a risk factor. We could think about other ways to enrich risk and have this same kind of benefit. So this idea of enriched risk cohorts has become sort of very well used in autism research and in some other fields of epidemiology that deal with rare diseases. And it's great because you gain back a lot of those advantages of a cohort study. You get sort of time-specific environmental information. You're not worried as much about the temporal ambiguity, although in autism, we're still not sure whether we should be looking at birth or prenatally or at conception, but you get better than you were when you just did case control. If we are interested in exposures, a couple of speakers mentioned that the prenatal window might be a particularly interesting window to think about. Well, if we're interested in that window, doing something where you have a birth or a pregnancy cohort and following them forward means that you have an opportunity for measuring biomarkers and potential molecular markers or other kinds of biomarkers, like you mentioned the eye tracking of Ami Klen's group or other things in very early ages. You have the opportunity to do that prospectively forward. Once you recruit a 5-year-old, you don't have the opportunity to get those kinds of samples anymore, whether they be molecular or other kinds of biomarker. And the thing that I'll show you a little bit about that I think is really cool is early life trajectories. And a lot of these larger data sets, you get some longitudinal information, often just about diagnosable conditions. But if you're interested in the sort of nuances of domain-specific trajectories that might be relevant to particular questions of the underlying etiology, then understanding that kind of deeper phenotyping and how that moves along in time is really critical. And that's the kind of thing that you really mostly only get by touching patients, by actually being there and assessing them and doing something like that. So these are kind of the advantages that you get. It's for sure at the sacrifice of sample size, although with this enriched risk, you may get to reasonable sample sizes, although you'll never be in the same ballpark that you've heard earlier, at least not often. So these are the 4. I was going to then just give you a little bit of example of the kinds of things that we do, and so, or that we have been involved in. Probably least of those is the case series, but I wanted to highlight that these are really great approaches to thinking about getting large numbers of cases where you can ask these questions and sometimes have appropriate sibling or other comparison groups. And so you heard about several of these. One you didn't hear about specifically that's been used a lot in genetics is the Autism Genetics Research Exchange, or AGREE, where they had traditionally taken very large multiplex families. So families where you identify a case that there's actually more than one case in the same family, and then you collect the family. You heard a lot about the Simon-Simplex plans and types of recruitment there. So identify a case, and in this case where there's a child that is unaffected in the same family. And then Ian, that many folks in the room have been involved with, is a registry where if you are either a parent of an affected individual or an affected adult and you want to engage in this kind of registry setting. All of these have all kinds of advantages. You heard some in terms of this FARI, the advantages to the person and the family for enrolling, but then it also has research advantages in terms of the kinds of connections that we can make. So those begin, though, and they're very convenience-based in some sense, because they begin with finding a case in the population. It's very different than taking a sort of general sample of the population. Case controls, as I mentioned, have some advantages. We are involved in one called the SEED, or Study to Explore Early Development project. This is a CDC-funded project that has 6 sites in the country. I'll show you those sites up in the top left. And this design was meant to be as population-based as you can get in doing this kind of work. So we start with birth and current residency frameworks. And then, so in Maryland, for example, we start with particular zip codes that people were born in. And then we reach out to those same places and we identify through vital statistics children that were born in those particular birth windows and in those particular zip codes. And we then recruit children in the same places that would be receiving some kind of developmental disability or ASD services through education systems and through healthcare systems and through self-referral. So we try to cast a very wide net, and that way we then have sort of population-based cases and controls from the same population. And then we do lots of assessments of those children to then in our own hands diagnose whether we believe they meet research criterion or not for autism versus typically developing. And this has gone through 2 waves of recruitment. The second wave will finish in a couple of months, and we'll have about 1,400 cases, about 1,400 population-based controls, and importantly about 1,400 developmental disability children, maybe a little less than that, that are non-ASD but have developmental disabilities of a very wide range. And one of the great things that we can get out of this, now you can see these are not sneezy numbers, but they're not the same kinds of numbers that you heard before, but this allows us to do sort of deep information getting. So we have extensive maternal or caregiver interviews, lots of self-administered questionnaires about the child's behavior and other kinds of domains. We get medical records from the labor and delivery, the prenatal care, the child's pediatrician, any specialty things they've done like genetics, and then we have folks who actually standardize, abstract information from all those medical records to populate a research database. So for things like, and I'll show you in the next page some of the domains of hypotheses, but for, say, a maternal infection hypothesis, we have information both from interviews with the mom and from medical record abstraction to sort of counterbalance information about what happened at birth. And then we get to see these children. So not only do we just have a 299 dot something kind of code, but in fact we do a full developmental evaluation. We have lots of information on them. You heard from some of the talks earlier today about the kind of spectrum and all the really lovely deep phenotyping happening with the 16P families and other kinds of families. We get some of that kind of look at all of our children, not just a diagnosis, but other kinds of things of how they're doing on IQ, how they're doing on other kinds of behavioral and cognitive performance. And we have a dysmorphology exam. We actually take a standardized positioning picture for all kinds of parts of their bodies, and we have medical geneticists who score every part of their body and come up with dysmorphology metrics about these children. So that kind of deep stuff is really exciting, but makes this much harder to do on many, many more numbers. And then we get an opportunity to do a lot of biosampling. So we get saliva samples, much like you heard some others. We also get blood samples on both the parents and the children, and hair samples on the children. And that means not only can we ask questions about genetics, but ultimately we can ask questions about environmental measures that you can get out of biosamples, maybe other kinds of biological measures like epigenetic things that I'll talk about in a little bit. And hopefully this helps us to answer two sort of domains of questions. The first is to think about distinct symptom profiles that might guide our etiologic work. So in addition to just yes or no to autism, what about some of these different kinds of domains and presentations? And then on the etiologic side, to ask questions amongst a bunch of different hypothesis categories that go from like infection to hormonal, GI, sociodemographic, etc. And importantly, genetics. And so that's not the only kind of case control studies that are happening in the U.S. A sort of mirror image in some ways to SEED is a case control study that's out of California called the CHARGE study. And folks now affiliated like Heather Volk and our center have been looking at the same kinds of questions in CHARGE, although many others, Lee Ching Lee and others, have worked with CHARGE quite a bit. And so this is just an example of one of the areas of environmental epi that has seen sort of shockingly consistent results over, I think, 9 or 10 epidemiologic studies now that have seen that air pollution exposure of some kind during pregnancy shows an increased risk for autism in the new baby. And so this is an example of that as measured by distance from freeway, distance to nearest freeway at time of birth. And so you can see here these adjusted odds ratios on the order of sort of 1.8 for close proximity. So that's case series and by extension case control, and there are certainly caveats of the dangers of that kind of design, and I hope I've made those clear, but also lots of advantages to what you can do when you can do a kind of very exhaustive approach with the individuals who you do recruit. As we said though, cohorts really are a place where you want to be for lots of additional advantages, and this enriched risk idea is one that can gain those advantages while still making it feasible to do something on a disorder that's relatively rare. And so many folks, both in the treatment and early identification world, as well as in the causal autism world, have moved to infant sibling designs. Infant siblings, as I said, are at increased risk because the family history, particularly the sibling history, makes it more like 1 in 5 rather than 1 in 68. And so that creates a much richer opportunity. And so we have one called the Early Autism Risk Longitudinal Investigation that's actually a network of 4 sites in the country. The network home is at Drexel in Philadelphia, and then we are a site. Lisa Krohn that you met was a site, and also Irva Hertz-Picciotto at UC Mind Institute is a site, or UC Davis Mind Institute is a site. And what's really cool about this is you gain all of these prospective birth cohort kinds of things. We actually recruited families who already have a child with autism as they were going through a subsequent pregnancy. So these are high-risk, soon-to-be infant siblings. And we did a lot of work with them during the prenatal period. So all kinds of things like interviews, assessments of the child that was already affected, then home visits at multiple times, biosamples at multiple times, additional interviews, and then right up close to delivery, we did a lot of work with them and actually carried around beepers so that we could show up on delivery day at their delivery center and get their placenta, get samples of the baby's first poop, get samples of breast milk if possible. Sometimes you have to come back after that to do some of those things. But it was kind of a really great experience because you're developing relationships with these families, including really being excited with them when these new babies come. So I think that part was really a lot of fun for our staff to be a part of, and really enabled the kinds of data collection that isn't possible in some of these other designs. And then importantly, we then followed them through to age 3. So we got to see how they're doing in terms of behavioral and cognitive and motor and all kinds of things throughout the first 3 years of their life. And so here's just a picture of them going into the homes. We did a lot of home environment work, actually taking dust samples in the home so you can measure kind of what things happening in the homes. A little checklist going through and checking what kinds of toxicants or sort of what kinds of cleaning chemicals and stuff like that are actually in the homes, those kinds of things. So really nice perspective of being able to see what's happening in their own environment as well as having them come in with us. And I should say, for the clinical assessments, and then once the babies were born, then they worked with our partners at Kennedy Krieger to get the evaluations of how they're developing. So it was a good example of that partnership as well. And so I mentioned all that's happening prenatally, and I just want to focus a little bit more on what was happening immediately postnatally. So cool that eventually we get to 36 months where we think a reliable diagnosis of ASD can be made, and was actually made by Becky Landa's group at Kennedy Krieger using sort of research criterion that is very rigorous. But we were able, based on her work and others, to think about early life, even earlier life metrics that might identify which of these children are at highest risk for on that trajectory towards an autism diagnosis. And these are things like assessments at 12 months where you can see kind of, response to name and eye tracking, or not eye tracking in the way that AMI does, but sort of whether they're going to try to look you in the eyes, things like that. And I'll say a little bit more about that in a second. And these early identifiers, we think, are really important phenotypes for etiologic research, because in fact, some of these children won't go on to get an ASD diagnosis, but they will go on to have reduced functioning in some particular domain. And so we actually think quantitative measures of development at 12 months and maybe at 24 and 36 and beyond are themselves relevant child-developed phenotypes for these kinds of questions. So I already mentioned a lot of these, but I just want to hit home, and I know it sounds a little bit like sort of beating a dead horse, but the difference between the sort of large number things and the small number things really comes down to all of this additional deep data that one can potentially achieve. And so all kinds of things, from the household stuff that I told you, to the things that we characterize about pregnancy in real time, to the things that we characterize about the new baby, and certainly all the kinds of phenotyping that we can do on these new children. And then in this study, we have biologic samples. The proband means the child who already had ASD that makes this family at risk. And then for the mother, we get blood, hair, urine, and saliva, placenta, cord blood, breast milk. For the dad, we got blood and semen during pregnancy. And then for the new baby, blood, hair, meconium, and urine. So all different kinds of biosamples that allow us not only to ask sort of biomarkers of what's happening in terms of phenotyping of the disorder, but also biomarkers of exposures and things like that. So I mentioned this early life period in green. The kinds of things that we can do with that data are look at some of this is from the Mullen scales of early learning, and you can see different kinds of domains that are measured by these scales, and this is at 6, 12, 24, and 36 months. And you can see the kind of heterogeneity you get across babies. Some are increasing and always improving, some are doing pretty poorly, going down, etc., or those kinds of things. What does this mean? How is this helpful? Well, Becky Landa's group had used data like this, not ours, but infant sibling data, to show that you can pull out classes of individuals by these early trajectories, and that those classes can be quite informative about how the children are about to be doing, and maybe have some etiologic relevance in terms of their differences. And so we think these might be good outcome classes for some of the causal questions that we want to ask. So Brooke Shepard in our group has been taking these kind of data and putting them together with some of the genetics work, thinking of these as an outcome for doing genetics-based research. And so it's a sort of way of taking advantage of that early life trajectory stuff and some of the genetic epi that we've been doing. And also taking advantage of her growing expertise and of those others in the center on thinking about construct or measurement issues, like a latent construct of these different kinds of trajectories that we don't directly measure, we infer through data like this. So she has to apply some statistical models to get to that. So there's kind of a methods development angle in all these, or methods application angle in these kinds of things. So that kind of gets at some of the sort of genetics and environment. I wanted to mention then an environmental example, and this really is where I think these kinds of data come in really handy, the fact that we have prenatal information that we don't have to get retrospectively. And there are many, many applications of this amongst the early investigators, but our group has mostly focused on the interplay between genes and environment, or the potential interplay between genes and environment, through a mechanism called epigenetics. And so I know that might not be a familiar term to a lot of folks, and I don't want to go too far into this, but I just want to say that when you think about the sequence of DNA, which is this kind of ACTG stuff, when we usually talk about genetics and we talk about the things that happened earlier in the symposium, often we're talking about whether there's a change in this A to a different letter or whether a whole section has been deleted or duplicated, those kinds of genetic anomalies that have been already associated with autism risk. When we think about how these things actually manifest into a protein and do work in the body, they go through several stages that require some kind of molecular regulation. And that molecular regulation happens by a whole bunch of other chemical processes. And so think about this as the fact that sequence is kind of like a faucet, that your sequence is telling your body and your cells which genes to make and at which time. And if you have regulation that says, turn that one on, I need a lot of that kind of gene, you sort of open up the faucet and the water flows. But at other times, you may not want that to be going on, and so you may turn the faucet off, and then you come down to just a drip or maybe off all the way. And so same sequence, same water that could be flowing or not, so that's the genetic part. The faucet are these epigenetic things that are molecules that tell you when to turn it on and off and where in the body. And so we think those are quite important because even for the same sort of genetic code, knowing that tight regulation of when to turn it on and off is going to matter. And in the genetics piece that you heard about in the very beginning, where there was those beautiful pictures of pathways and there's sort of synaptic pathways that are implicated. Well, one of the main pathways that's implicated from the genetic story in autism so far is chromatin remodeling and other types of gene regulation. The genes that are responsible for those kinds of things are the genes that look to be disrupted when we look at autism cases. So we think there's some reason to look at these kinds of molecules. So in our group, we've been looking at those kinds of molecules in multiple ways. Well, the first thing you'd want to say is, because regulation depends on where you are in the body, brains would be the thing to look at. But brains are tough to look at. You heard a little bit about why that's tough. Mostly that means we have autism-based research to do, right? And that means that you also deal with all the reasons that someone is no longer alive. That often means that you have brains that are pretty far along in neurodevelopment. Often they're adult brains. So there's sort of caveats to that, but that's where we are with the brain-based research when you need to have sort of pathological samples. But Chris Latticosta in our group has looked at epigenetic marks in brain samples and did find places in the genome where there's a type of epigenetic mark called DNA methylation, where the brains of controls look different than the brains of cases at that particular spot in the genome. So that's a hint that you could see those kinds of things in brain. We would rather think about, well, what do we know about early life and during development? Remember this whole idea of going at the beginning and following forward. And so with the kinds of biosamples you get in early, we can actually look in blood of babies, in blood of moms, and in blood and semen of dads at relatively or reasonably appropriate time windows. And so I mentioned to you that one of the cool things is looking at early life quantitative outcomes like at around 12 months. So one assessment is called the AOC, or Autism Observation Scales in Infants, that has been shown to have some predictive value about who will ultimately become a diagnosed case of autism. But as I said, I think it also is interesting to just look at the distribution of this kind of performance in general. Another one of these examples is SRS, and Dr. Chung showed you what the social responsiveness scales look like in populations. So I think on its own, this quantitative performance at 12 months is a reasonable thing to look at. So this is like, now imagine you're looking at how a 1-year-old is performing amongst these infant siblings. But we're going to then look at what that 1-year-old now's birth epigenetic presentation looked like. So a year before, when they were born, what did their cord blood DNA look like in terms of methylation, and how are they doing a year later? So baby to 1 year. And this is the kind of data that we see, and I know it's really hard to sort of figure out what I'm showing you, so I'm going to try to walk you through it quickly. Going left to right is a position on the human genome, and going bottom to top is the proportion of this type of chemical modification called methylation, and it can go from 0 to 100% for a particular sample from an individual. So if you think about a particular spot in the genome and a sample from a particular baby, right, then you can measure whether they have no methylation at that spot in the genome, 100% methylation, or somewhere in between. Okay? So each one of these dots is one of those babies and how much methylation they had. So this is like around 60%. And we didn't measure the entire genome, we only measured at particular spots in the genome. So that's why you see here's a scattering of data for that spot in the genome, here's a scattering of data for that spot in the genome. That's how you read across this. And the colors, blue versus red, are the children that did well versus not well on the AOC at 12 months. And so you can see these 1-year-olds that are doing well are the ones that are in the red, and the ones that are not doing so well are the ones that are in the blue. And for most of the genome, they look like this. There's no difference in what they look like as babies. But every once in a while you see something like this, where for a particular spot in the genome there's a separation. And so we don't yet know what that means, but we can identify these places where, where in the baby you have a spread of methylation, and that spread of methylation slightly corresponds to how they're going to perform 12 months later. So what we haven't figured out yet is, how do you interpret that? Blood is not the brain. Would it be even sharper if we had been able to look at the brain in those time windows? We're working with folks to sort of come up with how you compare blood versus brain in that way. But we think it's prospective evidence. We had no idea how these children were going to behave when we measured the methylation, or when we sampled to do the methylation, and then we see how they're doing 12 months later. So that was the baby. I think the interesting other thing is to think about moms and dads, and this is what other groups have not done as much of as in terms of the epigenetics, and even some of the ways that we're looking at the genetics. We had semen samples. So this is the actual, not exactly the gametes that created the new baby, because we did this after the baby was already conceived, but it's a profile that might be somewhat representative of the set of semen that was part of that story, right? And so now you're thinking about dads somewhere during that pregnancy interval trying to get as close to the conception time point as possible, and then what this baby's performance is at 12 months. So now we're going to look at what's happening to the methylation in the dad's DNA and what happens when that baby is 12 months. And here we saw a lot of changes. Across several parts of the genome, we saw where the methylation in dads whose children a year and a half later were performing poorly looked different than in the dads of whose children performed well a year and a half later. And it's the same kind of plot I showed you before, but there are more of these because there's more places in the genome where we saw this. And if I just sort of blow one up, you can see it's also for pretty big distances. But the same kind of correlation picture still holds. This is now the dad during pregnancy, and this is the baby at 12 months, and his methylation levels tend to be pretty sharply correlated with performance of the baby. And we saw that at multiple spots. And we also looked at mom, and I have not mentioned the folks, but Jason Feinberg, whose picture here, really worked on this one, and Kelly Bukolski, who in the first one and in this one, who's right here, did the analyses to see when you look at mom's methylation during pregnancy based on blood and this performance at 12 months, and you also see some places in the genome where you get this. And these are not typical. Most of the genome looks like this, where it's all pretty much together. These are, I'm pulling out the ones for you that are very rare but show these differences. So I bring that up not to say that we know exactly how to interpret data like that, although I think it's interesting that we're able to look at it in these ways. These are the kinds of opportunities that this kind of design allows us. So that was an example of an enriched risk where we can do this cohort study prospectively, and I mentioned there are other ways to do it. So preterm birth is also an enriched risk, so we are super lucky to be able to partner with Xiaobin Wang, who's here at the school and runs the Center for Early Life Origins of Disease. And before being with us at Hopkins, she was in Chicago, and before that in Boston. And when she was in Boston, she started something called the Boston Birth Cohort at the Boston Medical Center. And this originally started recruiting mother-infant pairs as soon as they came out of labor and delivery, essentially, and said, hey, can we enroll you in a study, and we're going to come back. But that consent allowed them to get birth biosamples and access to medical record data on them, and then later to ask about how their baby is doing. I think originally to ask about preterm birth and allergy, child allergy, and then has spread to other kinds of child outcomes. And we've been very fortunate to have received HRSA money to start assessing these children that are in this cohort for autism and developmental disability presentation. But we also can look in their EMR data currently just based on administrative records. And so we have kind of both designs, much like Lisa had with the HMO, the Kaiser, that we can use just billing kinds of codes, and we can go back and do individual touch kinds of phenotype assessment. So about 8,500 mothers, that would not be, or children, that would not be so great if we didn't have this enrichment, but there is enrichment for preterm birth. So we actually do have higher prevalence than you would anticipate otherwise, which might make this feasible for accruing enough autism cases. And we, in this sample, have lots of data collected once the child is born, and then we have biospecimens of maternal biospecimens at birth, cord blood at the placenta, importantly, and other kinds of blood samples at follow-up. So it allows, again, this prospective early life kind of biosampling. And these are the counts today. This is a still ongoing cohort of recruitment, so these numbers are likely to grow. But just to show you, you know, there's all kinds of information. This is specific to one of our doctoral students' project where she's thinking about infection exposure, and so she has as many as 5,700 participants in which she can ask that kind of data, and then she thinks about, well, what about also those with placenta, et cetera, et cetera. But even if you dwindle down to all the different things that we need just for her particular project, if you're basing it on EMR data, we still have almost 3,000. So I think this is quite an exciting opportunity for autism to do this kind of birth cohort in collaboration with the BBC. And so here's one example of a student in population and family health, Mingy Lee, who has been using these data to ask a question that has shown up a couple of times now in autism about the combined, or about the effects of maternal obesity or gestational diabetes or hypertension during pregnancy, these kind of related conditions. And she was able to really focus in on diabetes, both pre-gestational and gestational, and to also think about maternal obesity. And when she looked at those, she could characterize obesity and diabetes based on the med record data. And she could also characterize autism versus typically developing based on the EMR. So this does have the warts of administrative data because this is not yet updated with the phenotype assessment that we're doing now, but it allows this kind of look. And what she sees is similar to what we've seen in other reports that were more case control. There haven't been as many prospective cohort studies asking this question, but in this case, she sees that if you look amongst the non-obese just at gestational and pre-gestational diabetes, you see increased risk. If you look at among the obese and look at gestational and pre-gestational diabetes, you see even more risk. And so no matter which way you look at it here, no matter which way you look at it, you see independent effects of both gestational diabetes, pre-gestational, and obesity, but also joint effects. And so this is, I think, an ongoing thing that we're looking at in SEED, that we are able to look at prospectively in BBC and a couple of other prospective cohorts. So I think from those, I hope I've argued that epidemiology writ large, all these different designs play an important role in understanding autism, and that, you know, we can build an evidence base, and particularly these kinds of personal touch designs allow this kind of careful collection of both exposure and outcomes data. We pay special attention to measurement issues in a way that we're able to do in this setting. We're able to focus on these critical windows of exposure, particularly if we start at birth or before birth. And that really allows kind of important prenatal exposure information. So we have these deep phenotype measures. I could go on about molecular measures. I didn't even talk about the molecular measures of exposure as much as our group has been working on. So those are really exciting opportunities. It's not all roses and unicorns. There are certainly disadvantages, and that's why these other designs I think are also extremely exciting. The first of which is certainly selection bias. In case control studies, you often worry about that. Who is going to say yes to doing all this stuff, right? That's a big deal. And understanding that and working with methodologists like Liz Stewart and others to think about, if you had information on the full population, how can you analytically maybe get back some of that? But it's a tough question. Statistical power is certainly a concern. We sacrifice that to get these other kinds of data, and we have to think about the balance of that concern. And then generalizability. I think if you do a good job of population-based sampling and you're very clear about what your source frame was to begin with, I think you're in good shape. Where we get dinged is this enriched risk idea is certainly not a glimpse of the whole general population. It's the at-risk population. And so you have to be clear about what your target population is for doing enriched risk sampling and inference. We make the argument that internally, because our comparison group are amongst the high risks, those that do and don't have particular phenotypes, internally I think we can make a statement for causal validity and the kinds of questions that we ask. How that's going to relate to externally low-risk people is a real concern. And I guess my own editorial on that would be, I'd rather find some answers and then see how generalizable they are than be stuck without answers. But it's a legitimate thing to discuss as a field. And so I just want to end with reminding you that all of these that we heard about today, and many that we didn't even list, in my mind are all really important different perspectives to take on the way that we can collect data to answer these questions. As long as we remember the kind of pros and cons of each, and also to remind you that we're not really just thinking about studies that involve cause, but studies that think about the descriptive concerns and the services and policy, and then ultimately our goals are to think about how these things can inform prevention and intervention. And I tried a little experiment to come up with the different studies that were going to be discussed today and kind of the pros and cons. So to try to go a little bit more in depth than this cute little up and down part. I don't know how far I got, and you might want to disagree with me, and these are, I think, going to be on the web, so you can read this and then write us in and say, no, I totally disagree. I would love that. But I tried to create a sort of heat map where red is a lot or very hot, and very deep blue is cold or not at all. And so I think it helps you to see that things like sample size, temporal clarity, maybe policy and service delivery kinds of things, some of these registries and HMO things might do really well in those. But when it comes to getting enough biosamples and maybe environmental data or phenotype quality, that sometimes these personal touch ones do better. And I think the thing that I would point out is if you look amongst the rows, no single row is all red. So no one of these designs is a terrible idea. On the contrary, though, no single row is all green. So no one of these is going to be the only solution. And I think it's by messaging with each other and hearing from each other that we talk about how all of these have a role to play and a place to improve the science and thinking about autism, I think would be better for the field. So thank you all for spending the time to do this with us today. I cannot go without thanking all of our research group personally, but also our colleagues at Kennedy Krieger and my colleagues in the Center for Epigenetics and our colleagues in the Biosample Repository here in the Bloomberg School, all the collaborators of these studies that I'm lucky enough to be involved with, and the people who are nice enough to fund us to do some of this work that I think is so important. And then last but not least, all the folks involved with the center here at Hopkins, and all of the folks that cared so much about Wendy, that they have given their time and financial support towards this endeavor, and continue to do so. And then at the very end, in this big morass of folks that's a little bit dark on this slide, there are two very, very special people that you have to blow up, and that's Michelle Landrum, our center coordinator and sort of everything extraordinaire, who helped put on today but really keeps the center running, and Jamie, who is our coordinator for SEED and really also helps keep the center and the internships and all kinds of things. These two are really the heartbeat when you're on the second floor around here and you see the Wendy Klag Center offices. They're the ones that are really making all that go. So thanks again to everyone, and please join us downstairs because there's so much more work going on here than any of us could cover, and I would love for you to talk about it with each other over a snack. So thank you all.